Everybody to the grand finale of the BioAfrica Convention 2021. This is our closing session, closing plenary, and as is now becoming our tradition, we close the session with myself, the current president, and I mention current because it's not a hereditary post, it's an elected position, so there may be other dialogues with Global Biotech Leaders hosted by other presidents that will come after me. So I think I am very fortunate this year to be hosting a very, very distinguished panel, most of whom, if not all of whom, are my personal heroes. And I will just quickly introduce them and go across the screen. I'm assuming that the delegates are seeing the screens the way I'm seeing them. At the top left-hand corner is Sir Richard Roberts, a Nobel Prize winner for medicine in 1993. In the middle is Dr. Anthony Ndifo, who is a J&J working on preclinical development. On the right-hand corner, top right, is Sir Richard Roberts. I'm not Sir Richard Roberts, my apologies. Lawrence Banks. I've just given him the knighthood, you know, because it's just been so good to me this week. So it's Dr. Lawrence Banks from the International Center for Genetic Engineering and Biotechnology. Below him is Dr. Yemi Onakunle from VLP Biotech in Los Angeles. And next to him is my old friend, Dr. Dennis Leota from Emory University in Atlanta. What puts this distinguished panel together is their mission to make this world a better place through biotechnology. And a lot of the work they've been doing, a lot of the work that they are going to be doing in the near future is going to be focused on contributing to the theme of advancing Africa's manufacturing capabilities through innovation. And they will share, each one of them will share their perspective, will share their unique stories, which I cannot wait to hear. And I will start with my friend who is actually sitting right next to me. And it was going to be the guy on the right-hand corner at the top that I was going to ask. That's how I had written it last night. I didn't know that it was going to be Dr. Lawrence Banks. If Dr. Lawrence Banks can start maybe just to give us a perspective on the theme of the convention. Thank you.
Thank you very much, Nhlanhla. This has been a tremendous 2 days. Your team here do a fantastic job in putting this amazing conference together. And I think it's one of the few that I've ever gone to which has a real aim in mind, and that is to ensure that we can really deliver the fruits of modern molecular biology and biotechnology to the populations of Africa. And I think everybody who comes to this meeting really shares that vision. And so it's very pleasing, it's very enjoyable to see such a common focus amongst all the participants. And you know, I know it's a huge challenge doing this as a virtual format, but nonetheless it's still incredibly successful and incredibly enjoyable. So that's my take on the meeting. I think you're doing a tremendous job, and I would just say just please keep doing it. I have prepared 4 slides. Would this be appropriate? Okay, I'll share my screen. Okay. So I think one of the things that really comes out of this meeting is collaboration. And a fundamental aspect in science is collaboration. As scientists, we work together, we work in teams, we have interactions that span the globe. But I think really, if we are going to meet the SDGs, particularly for the African continent, we need not only to just collaborate as scientists, but we need to collaborate as institutions, organizations, and as countries. And it's through these partnerships that I think that we're collectively really beginning to make a difference. And I just want to highlight one partnership that ICGB has been involved in this last year. It came with COVID-19. It was initiated by Rich Roberts when he's on the Council of Scientific Advisors of ICGB. And when we had our CSA meeting last year, in April, May, he said, he really raised the issue, which is absolutely fundamental, and that was getting diagnostics to low and middle income countries for COVID-19. Clearly, the standard technologies were not available in sufficient quantities, and they were clearly far too expensive. And so ICGB, with New England Biolabs and the Bill and Melinda Gates Foundation, teamed up to develop a program for trying to get a cheaper, simpler form of diagnostic for COVID-19 to labs in Africa. I have to say that was incredibly successful, and we rolled this program out in Nigeria, Cameroon, Ethiopia and Kenya, and now we're moving into a phase 2 program, which is going to move this diagnostic obviously further within Africa, within those original 4 countries, with appropriate regulatory authorization. We're expanding the network into additional African countries. And I think one of the things that's also incredibly important, and Rich may also add something to this in his presentation, is that it's critical to assist in the development of local production facilities to ensure sustainability of these diagnostics and future therapeutics. And one thing that I'm incredibly excited about is that through this program, ICGB has also been able to act as a reference center for trialing new diagnostics post-COVID, because clearly we need to look beyond COVID and for ensuring that such technologies can be made relevant and functional in a low-resource environment. And finally, and I think this really goes hand in glove with the whole concept of the BioAfrica Convention, is that we have to really make technology transfer easy and practical for countries across the African continent. And one of the things that we're actually also very keen to work with them on is to develop an exchange platform for identifying IP which is available and which can be transferred to African countries to ensure that these technologies actually reach the people that really need it. So with that, I'll stop. Thank you very much.
Thank you, Dr. Banks. I do not want to explore the presentation further. I just want us to jump straight to Dr. Tony Ndefo, just with his opening remarks, because I suspect when you talk about technology transfer to the continent, particularly within the portfolio of the ICGB, one of the critical steps in such a transfer modality would be to do some clinical development work. And I'd love to hear what Tony has to say about the infrastructure required to prepare the continent for accepting technology at that stage of development. Tony, over to you.
Thank you, Dr. Msomi. I hope you can hear me well. And it's my distinct pleasure to be on a panel with such esteemed scientists and colleagues. I know we talk about biomanufacturing, which is extremely critical. But I think one of the things that we got to think about is that before we even get to the stage where we are making any of those medicines, vaccines, either vaccines or any biologics, is we have to first of all go through the hurdle of safety, and that's it could be non-clinical safety or just clinical safety. Thanks to the COVID pandemic recently, There has been a broad acknowledgement and awareness in the public of safety issues around medicines. But you know, what is less hidden, what is more hidden or what is less obvious is what goes into many of these new medicines before they ever even get into humans, and that's in the preclinical space where I operate. And for medicines that will be that would require biomanufacturing, so you're talking about vaccines, you're talking about maybe protein or peptide related medicines. These have a special, when it comes to non-clinical safety testing, they have a special pathway that they take, mainly because one, they are proteins, which means that they need to be injectable, so they need to be given in a very, in ways that are formulated. If we figure out a way to give them orally, the formulation has to be very, very distinct and specialized. And so the challenges of doing this is finding, first of all, a preclinical model that you can use to test any of these medicines. If you know, those of you who are in the vaccine space, you know, monkeys are our next neighbors in the evolutionary tree. Monkeys and rabbits tend to be used quite frequently, but if you expand that to more broader range of biologics and peptides, then you can, you know, in many cases, it's still non-human primates that are used in assessment of these components, and it is quite an expansive and also an expensive endeavor, because you're not just, when you talk about testing, we are talking about, in many cases, running trials that can run up to a year. And we start with, you know, short-term trials to support initial entry into humans, but those trials can actually, in this trials and non-clinical models, can run up to a year to support the appropriate trials that are also going on in humans. So these are, you know, in themselves expensive endeavors. These are in themselves specialized endeavors, but these are also activities that, in a very smaller scale, are going around all across the continent. We have multiple scientists and researchers who are running labs, and using animal models. So it is not a stretch for you know, any of those institutions to develop activities, to develop capabilities that can be become GLP compliant in the long run. GLP being good laboratory practices. Compliant, that can then be able to adopt, to be able to use to run some of these initial trials that are required by regulatory agencies to support any trials, including even assessing, you know, going to biomanufacturing, assessing the purity, the impurity profile, and you know, endotoxin and all those things that are required. So we you know, at the moment you can think about it and say we don't have the full capacity to do it, but it's not, we are not far off from being able to piggyback on what we have and building on that and pulling resources across the entire continent to set up facilities that can support many of these early drug development activities, and also you know, end up reducing our costs substantially. Because if you take any of these studies, you want to run them abroad, you want to run them in UK, you want to run them in Europe and the US, they can run you in millions of dollars. But you know, my colleague Yemi would know that you can go to China and get a fraction of the cost, which means, because the labor cost is really what is critical in this, and which means if we set up such capabilities in many of the African countries, those labor costs can actually be reduced substantially, and further on reducing our cost of development and entry. I'll pause there.
Thank you, Tony. I think one of the issues you've just raised, which is critically important, is the issue of not aping or mimicking business models that have been done elsewhere. You alluded to a possibility of pulling in resources at a regional level, say across the continent. You're looking at a preclinical infrastructure, say in West Africa, East Africa, and there's a pooling of resources in Southern Africa, because individually these countries or companies may not be able to do it all by themselves because of the cost. Not only the cost, but also the availability of expertise. And that is a very, very important point in developing strategies that would work in ensuring that Africa has enough supply or has security of supply for vaccines for any future pandemic. And I think that leads me to our next panelist that I'd like to invite Sir Richard Roberts. I know he insists on me calling him Rich, to share with us a model that we found fascinating of New England Biolabs. A lot of this convention has been about biotechnology and entrepreneurs and financing, and we've not explored whether there is an alternative model where we could develop products through a human-centered or a human-driven approach to innovation. Thank you, Sir Richard Roberts.
Okay, well, I really want to talk to you about two things, if time permits. The first is a project that we're looking at the moment at New England Biolabs, and building on the way in which we became a successful company, we're thinking we would like to try and do the same thing in Africa. We would like to start a company in Africa that eventually would be owned completely by the Africans who took it over. The idea is to start on a small scale, making enzymes, making oligonucleotides, and making test kits for COVID or for other diseases. Our intention is to finance this completely, to train the people, and this would be a donation. It is not something that anybody's going to have to pay back. The company would mirror what we do at New England Biolabs. We're a profit-sharing company, so the employees share in the profits of that company. We started small and expanded as we got sales, and we believe that something like that would be eminently possible in Africa. And to give you an example of the kinds of things that we make enzymes that are necessary in order to make vaccines, the mRNA vaccines. So many of those enzymes that the companies use, we would transfer that technology, allow people at this company in Africa to do it themselves and to make other things that are necessary for molecular biology experiments. And we've been told oligonucleotides, for instance, are something that are often difficult to get. We would set up an oligo facility. And of course, the test kits for COVID would be an ideal first product on something that you might spread and could be quite successful, could be quite big. So that's our intention. We're looking at the moment to find the right country to do it in. And we're also looking for the right person to run it. We're looking for a CEO who is not interested in just building up a company and then selling it for profit, but rather someone who wants to set up a company and help Africa in the way that we do. The other thing I would like to talk about a little bit are GMOs. And I think one of the things that is really critical in Africa, if you're ever going to feed your population successfully, is to adopt GMO technology. And I think the Europeans have done you a tremendous disservice by trying to stop countries from developing and using GMOs. And the nice thing about the methods, the modern methods that were developed some 30, 40 years ago, are that they are simple. They can be done very adequately by African scientists, and in fact have already been done in labs in Africa. They can have a tremendous impact on what goes on. One example, for instance, is bananas in Uganda were suffering from a major disease, from Xanthomonas wilt. African scientists in Kenya came up with a genetically modified banana that was resistant to Xanthomonas wilt. There are no naturally occurring varieties of banana that are resistant. And so this is something that you could only get using GM technology. This is something that should be spread around in Africa. Governments need to be taught how easy, important, and safe the GM products are. And we need to make sure that that gets going. If you just look in South Africa, you're growing BT corn there, which is resistant to the fall armyworm. In other African countries, sub-Saharan African countries, fall armyworm is causing major problems. And I think when thinking about this, you should be thinking about Africa and not Europe. You know, the hypocrisy in Europe over GMOs is quite appalling. I don't know if you know where human insulin comes from to treat diabetics. It comes from a GMO. You don't hear the anti-GMO activists talking about that. Cheese that is made in Africa uses GM organisms in order to ferment it properly. And perhaps the biggest single problem I have with the hypocrisy is the animal feed, the feed that goes to the cows, to the sheep, and so on. This is almost all GM modified soybean. Millions of tons are imported into Europe every year. GM crop fed to the animals. Why aren't the animal rights activists so concerned about the dangers? Well, because there are no dangers, that's why. In the 30 years that they've been producing GM crops, there has been no problems whatsoever. There is not one example that is scientifically credible of a problem. With that, I will stop, and I very much hope you will talk to your governments about the value of GMO crops. Thank you.
Rich, and I think this is a panel of biotechnologists. So I'm pretty sure we all are enthusiastic about the possibility of using genetic engineering and biotechnology to ensure food security in the continent and across the developing world, particularly with regards to the types of foods that we eat in this part of the world. And I think I've exhausted the top panel, not because they are top in relation to the bottom panelists, just on the screen. And I'd like to move on to Dr. Yemi Onakunle, who is an inspiration. He has worked for a very long time in biotech and has been involved in a number of major transactions while he was at Celexis in Switzerland, and so would understand the licensing deals that could give effect to the dream of advancing biomanufacturing in the continent. I'd like him to just touch on that and maybe end off by sharing with us his current work in his own company, VLP Biotech. Yemi?
Yeah, thank you. Firstly, I'd like to thank the organizers for putting together this really esteemed conference and also for inviting me to be a part of it. I would say that it's very endearing to me because despite all the efforts that we put here in the West, our real focus is how to figure out translating a lot of the knowledge that we acquired here to advance the development of the African continent when it relates to biotechnology. As Dr. Msomi mentioned, I have come through a traditional biomanufacturing background, having worked at companies like Lonza and Fujifilm Diosynth, both in the US as well as in Europe, and most recently as Chief Business Officer at Celexis, which I exited at the end of March of this year, having worked there for 10 years. And what's clear to me is that there is a rapid evolution of technology in relation to biomanufacturing of next generation molecules like antibodies, proteins, and now we are seeing a new class of molecules that are being introduced to harness the body's natural mechanism of making these molecules, which is using mRNA. What's clear to me is that it takes a major ecosystem to come together, which will comprise a lot of financiers, a lot of major pharmaceutical companies, universities, and grassroots scientists, all working together in order to be able to advance these technologies, because you plant the seeds early and reap the rewards later after lots of experimentation and failures. But ultimately, that leads to success. Yesterday, I was on a panel, and what was clear to me in all the discussions we had is there is definitely a genuine interest among scientists, both on the African continent as well as abroad, to figure out ways to bridge that gap. And one of the questions that came up is, how do you actually de-risk the investment in such science? And one thing I want to mention is, there's a general saying that if it doesn't make money, it doesn't make sense. In this case, I don't think we can take this approach. I think we really have to look at it that even if it doesn't make money, it does make sense. And the reason why it makes sense is actually because of some of the things that the earlier panelists have talked about today, in saying that we want to make investments in Africa to make Africa self-reliant, but importantly, to build the infrastructure. And this is where I'm really looking at this as well, is to figure out ways that we can build the ecosystem within the African continent so that they become more self-reliant and can actually have a mutual partnership of equal value between the African continent and the rest of the world. Now, I mentioned earlier that I recently decided to embark, and actually a lot of this was a result of the recent years that we've had, where I thought, well, the only way I can really start to gain the sort of leverage I need is to go forward and walk the steps that I keep talking about. And this is why I've decided to really start channeling this entrepreneurial spirit into developing and hopefully being successful in developing technologies and companies that will be relevant to Africa. The first company that I'm working on right now is a company called VLP Biotech. And in fact, VLP Biotech has been around for the past 20 years in San Diego, working on vaccines. I've been a board member of this company for about 10 years. And my interest in this company was because of that. They were developing vaccines. The challenge with that is nobody was interested in vaccines 10 years ago. And the reason nobody was interested was because actually it didn't make money, so it didn't make sense. Today, we know that was a big mistake, because actually, when we talk about vaccines, and I can share some quick slides here, just bear with me. When we talk about vaccines, I think we can recognize that vaccines have really evolved significantly over the years, where you're really trying to harness the potency of the vaccine by looking at the authentic molecules, but trying to improve the safety by going to key determinants of the immunogenicity that allows for protection. However, what's been clear from the recent pandemic is that it takes a lot of investment in order to be able to develop these molecules that are supposed to protect us from disease. And so we are seeing new technologies that have been highly successful, thankfully, but unfortunately not readily deployable because of inherent intricacies associated with their manufacturing and delivery to the regions that need it the most. So what we are doing at VLP, and in fact, I had actually put on here previously a prescription for what we need to do, but actually I think I would like to think of it in terms of what are the must-haves for a world vaccine. And for a world vaccine, I think we definitely want to maintain the protection and efficacy. We want it to be compatible, however, with our existing infrastructure. For manufacturing, it should be readily scalable and deployable, stable. Importantly, no requirement for cold chain, because we have to deliver these across several countries on the African continent, many of which do not have suitable infrastructure at the moment to handle such cold chain requirements. And importantly, we need low cost of goods. So in fact, our platform is what we see here. It's called a virus-like particle based on a species variant of the hepatitis core antigen. And what we're able to do is potentiate single epitopes that are safe, and we'll be able to engineer it into these platforms, which you see the monomer here, that then allows us to generate a very strong immune response for protection. And I can tell you that these particles can be expressed in E. coli, so they're very easy to make. And importantly, they're very stable, even at room temperature. Today, we have two active programs, one of which is actually on a head-to-head comparison with the existing vaccine for malaria, which was approved recently by GSK. So we would see the outcome of that before going into the clinic, but we also have another candidate for chronic hepatitis B. So with that said, I just want to end up by saying that to advance Africa's biomanufacturing value chain, we really have to think about what are the critical resources and how can we access them. Expertise, financing, we really need to think about clinical trial networks, the regulatory framework, as well as distribution channels, recognizing that we have over 54 countries in Africa, all operating independently. Yet we are moving in the right direction with our free trade area that is being explored right now and has a goal towards 2063 of being a fully integrated market. Importantly, I think we should start thinking about, and I was glad to see in some of the earlier talks that we're talking about win-win partnerships across Africa and the rest of the world that can create value for all involved. We should remember, Africa is 1.3 billion people. So if we frame this the right way, companies, even Western companies, should see Africa as a very attractive market to invest in. And finally, I would really like us to start thinking, how do we actually interact with governments in all the different countries? Because not having access to biomanufacturing, it becomes an existential threat to the continent. There should be an interest from all parties to be involved. And with that, I would like to stop my talk. Thank you.
Thank you for that expansive perspective. The last comment that I'll invite goes to my great buddy, Dr. Dennis Leota. Just a little bit about Dennis. We've had a very long relationship, but for me, his immense contribution to contributing towards converting what was previously a death sentence to African populations, especially in this part of the world. I come from Durban. This convention is actually being hosted in the city of Durban, which at one point in time was the epicenter of HIV/AIDS. It killed pretty much most of my generation. And so when I first met the guy who converted the AIDS infection from being a death sentence to being a chronic manageable disease, it was a personal moment. I'm sorry, Dennis, I have to put you on the spot. I'm not trying to embarrass you, but I'd like you to share some perspectives, particularly around innovation. Earlier today, we had some discussion on manufacturing value chains in the pharmaceutical industry. And I recall some conversation we had some years ago around the continuous flow technology for pharmaceuticals as one of the newer innovations that could be adopted by countries if they wanted to break free from the yoke of the Far East manufacturing juggernauts. I'd like you just to share a bit of your journey from an innovation perspective, but as we say in this convention, is advancing the manufacturing value chains through innovation. Over to you, Dennis.
Thank you very much, Nhlanhla. And I have to just clarify your remarks because as all our panelists know, drug discovery and development is a very complex process, and hundreds, thousands of people, all at different times in the process, make pivotal contributions. So I reject the idea that I single-handedly did this, because I know so many smart, capable people who really made very important contributions along the way. I think it's important for everybody to understand that we talked about teamwork and collaboration, not just on a laboratory scale, but a multinational scale is going to be needed to address many of these things. So let's thank you. I'm flattered, but I do want to put my contributions in perspective. And I'd like to start off talking about innovation, and I want to share one of my screens and just spend a moment talking about this. Let's see. That's probably the wrong screen, huh? There we go. So for about the first 2 decades that I did drug development, I didn't fully understand the process. And I wanted to try and, in a very simple fashion, try and describe the process by looking at 3 variables: risk, value, and cost, to try and make a point about what I intend to talk about today is more harnessing innovations and creativity from the academic sector. So many of my colleagues in the academic sector will have a wonderful idea. They'll wake up in the middle of the night and they'll say, I have this great idea, and this is going to change the world. And sometimes the ideas are quite excellent. But from an investor's perspective, those ideas are 100% risk. And so what we typically do is we do a series of experiments to de-risk those ideas. What many people in academics don't fully appreciate is that this de-risking process is not a linear process, at least when it comes to value. It's a step function. And so what we have to do is reach or move our technology to value inflection points. And at each value inflection point, we've now added value to the technology. And the good news about academics is that we can tend to do this at relatively low cost, because the really expensive parts of drug development are the clinical trials. So what we should try to aim for is that sweet spot where we've lowered risk and added value, but done it at a relatively low cost. And I want to show you a model that we developed at Emory that I think could fit the bill for one of the regional centers that you were talking about earlier. And this is a model I mentioned briefly in my presentation. We call this process Drug Innovation Ventures at Emory, or DRIVE. And the purpose of DRIVE is to serve as an industrial partner, in our case, to Emory, but in the case of Africa, it could serve many universities in the region. And it would provide a unique combination of scientific drug development expertise commonly found in pharma and biotech environments, and management oversight expertise like that found in commercial biotechnology and pharmaceutical companies. And so what DRIVE does is they identify therapeutic opportunities, they in-license candidates, they manage their progression into the clinic and through the very early stages of clinical development. And it can contract with various partners. In the case of Emory, we have something called the Emory Institute for Drug Development, which is shown there in blue. And they work together to advance technologies to new value inflection points. I think one of the interesting points, just to not dwell on this too much, is to maintain its fiscal sustainability. DRIVE pursued a mixed portfolio of need-driven projects with small markets and others that have substantial and well-established commercial markets. And the idea was that the well-established markets could provide revenue streams that could be plowed back into the operation and allow us to move forward on other important areas whose markets hadn't yet been established. And one of the interesting examples that emerged from that was, for several years, we have been studying single-stranded RNA viruses, and it's estimated they account for about 80% of the world's viral burden. And these markets are, with the exception of hepatitis C, there are no drugs to really treat single-stranded RNA viruses. So we were working on trying to identify broad-spectrum antivirals for these single-stranded RNA viruses. And in the process of doing it, we came across a compound that I mentioned in my talk called molnupiravir, which is now in phase 3 clinical trials and hopefully will receive emergency use authorization to treat COVID-19 infection. So this was a serendipitous case of working in an area where there really was no market for a lot of these diseases, the various equine encephalitis viruses, and a small market for dengue and chikungunya and many other viruses, Rift Valley fever, and so by working in this area, we were fortunately in the right place at the right time, and this enabled us to come up with something that may actually address this. And I think such a regional center in Africa could support and a lot of the innovations developed in academic laboratories and help to translate them going forward. I'll mention two other things briefly. One is an effort we started up several years ago with the acronym AHIA, Advancing Healthcare Innovation in Africa. And this was an effort on our part to support health innovations and technologies by advising, educating, and training innovators in the business and legal aspects of the healthcare sector. And we have workshops yearly where we invite African scientists to come and join a team of students and faculty who work with them to create development plans for their technologies. And then we consult with them as the technologies progress. We've also, during the pandemic, initiated a series of webinars In this one that I'm showing here, we invited 3 graduates of our workshop to talk about their innovations. And these webinars have been very well received because they are the innovators from Africa telling their story. And this has, I think, been very helpful. The last point I'll make is what Nhlanhla mentioned earlier, which is this idea of incorporating continuous flow manufacturing capabilities into traditional biopharmaceutical manufacturing. This is, I think, very critical and very timely. We talked earlier about food security, but I think it's a source of concern, or should be, for every nation in Africa, that there aren't really any robust pharmaceutical manufacturing capabilities there, or certainly not enough. And if we use the old approach of batch manufacturing, it would be very hard for those facilities to compete with China and India. But if we incorporate new technology like continuous flow, which is much less expensive to set up, has a very small footprint, and can be distributed regionally around, there are excellent opportunities to leapfrog past the traditional batch approaches. And I think the analogy to cell phones versus landlines is fairly legitimate here. So I think there are many opportunities to do that, and we have to, as was suggested, I think, by many of our previous speakers, we have to do this collectively, not competitively. We have to learn to work together with our colleagues in the commercial sector, with investors, and with governments. So with that, I'll stop my remarks. Thank you very much.
Thank you for that, Dennis. I think what I'm going to do now is to ask a question that goes directly to the theme of this year's convention, seeing that this is our last session of the convention. I want to probe certain elements of our theme, which is advancing Africa's biomanufacturing value chains through innovation. And really, I want to probe the notion of value chains. And my mind picked up when Dennis mentioned cell phones, because cell phones are a good example of a value chains driven product. And that often where we are in this part of the world, we tend to be obsessed with a notion of innovation that tends to be dogmatic. And we have this linear model that is based on, let's say, a traditional concept of discovery science through a value chain, through clinical development all the way. And this is just a provocation to yourselves to say that if we want to think about biomanufacturing's value chains seriously through innovation, what other types of innovations should we be exploring? Yemi, I know, has worked in Slaxis and has done a lot of licensing deals that are really geared towards ensuring that you don't have to do the entire value chain in terms of development. I know ICGB does a lot of biosimilars, which are matched to the public health needs of the continent, and there is another opportunity for exploring value chains and exploring innovation in a different context. And I would like to also invite Sir Richard Roberts with regards to the diagnostic test, for example, for COVID-19. But everyone is invited, and this would be, let's say, the last question of this session. So I would like to invite individual panelists just to probe the question further, how is it that how can we advance this much needed biomanufacturing value chains through innovation, and thinking of innovation outside of the usual traditional, almost ideological sense of a discovery line from discovery to the product. Maybe I would like to start with Dr. Lawrence Banks, because you're next to me.
Thank you, Nhlanhla. I think the innovation has to come in terms of being innovative about what we decide that we really want to produce. Clearly, in the current situation, it's vaccines for COVID-19, but we're going beyond that, and we're trying to look at products which are going to really make a difference on the ground in Africa. And there's an awful lot of them out there. We don't have to reinvent the wheel. I'm taking the approach of the biosimilars because they're there, the technology's there to do it. What you need is the innovation to actually generate the manufacturing and production capabilities in sites in Africa and subsequent marketing. My major concern is that, and I think one of the earlier panelists mentioned this, it doesn't have to be profit driven. I think it's very important that you can provide medicines, diagnostics, which can reach the people on the ground who really need it. And so it does need an investment. But I think when you talk about the innovation, it's trying to break away from saying we have to develop a completely new therapeutic for something, when in a lot of the cases, to get going, to get the whole process moving, you go for something which you know is going to be a sure winner.
Thank you. Thank you for that comment. And I was going to ask the same question, because I know Yemi is quite passionate about thinking in an innovative manner around business processes that would bring healthcare products to the clinic in the continent. Just a bit about that, Yemi.
Right. Actually, I couldn't agree more with the comments just made. I definitely think that we have to be selective in what we mean by innovation. We don't have to develop new drugs if they're already made. So the question of, if it's not broken, don't fix it, that applies here. But we have to also be smart. We talked about de-risking investment. There's a lot of technology already that exists that we can start to implement. The question is, where do we start? One thing that we have explored in the past is a scenario where you, at least whilst you're still building the infrastructure needed to take things from the start of the value chain through to the commercialization stage and the market, that you develop, or the design of those processes can be done or that already existing, and then you have a tech transfer model where you just implement a process on the continent. And I think that can work because the technologies, as have been highlighted, they are available now. We can actually leapfrog from the traditional confines of a batch process using things like disposable technologies, using things like continuous manufacturing workflows. We've actually also thought about how you could make these more mobile, such that you're able to install installments in key hubs on the continent where you've already built critical infrastructure. So I certainly think that there's a lot we can do on the manufacturing side without redefining the wheel. One last thing I would mention, however, is to think where we are today. Because when we're talking about biomanufacturing, you can think of it from the point of view of the API, which is the drug substance, and you can also think about it from the point of view of the drug product, which is the field product. And I think as a in investing into the drug product manufacturing at this point, because now you can really amortize that across multiple inputs to deliver drugs to the continent. I think that's a good starting point, because again, we have to think about how do we bring up the ecosystem and infrastructure to be at the level where it can be self-sufficient to supply the entire continent. I probably stop there.
Thank you, Yemi. I want to move on to Rich. I just want to probe a bit further, and this is where I'm coming from. There seems to be this wave or hype around monetizing biotechnology innovation, a hype around making a profit. But it seems to discount the fact that the real engine of innovation is intellectual capital and human capital. And so I want to explore the business model where biotechs that are highly productive are actually co-owned by the inventors, not in the usual way where inventors are rewarded for breakthrough inventions and they get paid big checks, but in the way where people build entrepreneurial ventures with a lot of human capital that acts as a collective. Maybe you can just share some perspective on that, Rich, particularly around how New England attempts to achieve that as we are looking to establish a base in the continent.
Right. Well, I'm a great believer in starting small. And New England Biolabs started with 3 people in a basement, actually underneath a hairdressing salon. And within 1 year of getting going, they were selling several $100,000 worth of enzymes. And I think one of the things that it's all very well to think about the top end and delivering medicines and so on. But I think one thing that's important is to remember that the supply chain leading into all of these businesses is extremely important. And this was where New England Biolabs saw an opportunity because we are at the bottom level of the supply chain. And by supplying the right reagents, you can make enough money to become successful. But money has never been our objective. We're not interested just in making money. We're interested in getting a good business going, providing research resources so that researchers can do their business better and not have to pay an arm and a leg for it. And we've always found that we've been able to make enough money this way that we can then support our own internal research efforts. We have more than 100 people at Biolabs who are focused on doing research. And out of this, the C-LAMP technology for diagnosing, for looking to see if you have COVID came. That is something that can be expanded into many other diseases. I would also say that by ensuring that research continues, one can make breakthroughs that could be really useful. And if you look at the vaccine indications, mRNAs are terrific. I mean, these make excellent vaccines. They're easy to produce. You can change them very quickly, but getting them to people is difficult because of the cold storage requirements and so on. But what about the African scientist who came up and said, I know how to make mRNAs that will travel at room temperature. You can just put them in a box and ship them wherever they have to go. That would be an innovation which would be terrific and would have very widespread applications. And I think it's eminently possible to set up a business in Africa where those kinds of innovations are possible. One of the great things about Africa is you have such genetic diversity that not only do you have the very worst people who end up as dictators, but you end up with the very best people like Nelson Mandela, who have fabulous ideas and are really great. And we need to find those. The good marathon runners always came from Ethiopia. Let's find the good researchers wherever they're located in Africa and give them the opportunity to make the innovations and to make everything possible. And if you do it in the way in which we did it at Biolabs, we share any revenues that we get with the employees. So our employees, no one wants to leave. Everybody loves Biolabs because they get a big profit sharing check every year. And it just establishes a spirit within the company where people want to work together, they want to help one another. Whenever you ask for a volunteer, you usually have 20 sign up within 10 minutes.
Thank you. Thank you, Rich. I'll move to Dennis, and I promise I'll not embarrass you this time around. Um, you mentioned one or two examples of how Drive operates, and it seems to me that one of the best or the best possible sources of innovations and of innovations that would start to turn the tide for the continent would come out of our universities. I think in an earlier session, I mentioned that 60% of the African population is under the age of 35. That's a massive number of young people. And what it does, it means that there is the world sitting there as latent intellectual capital or human capital. And maybe what I'd like to hear from you as a parting shot is how universities or how research institutions in Africa could start to harness this immense intellectual capital towards providing solutions through innovation, and how it's been done at Thrive.
Yeah, so you're right. The potential is enormous, and it would be criminal to leave it untapped. Africans in the more than 2 decades that I've been there working with colleagues like you are not only very smart, very capable, very hardworking, but they have a special zeal associated with addressing local problems. And they really love the idea of African solutions to African problems. The West has tried for centuries to come in and tell people there, this is how you do it. And of course, we failed for centuries by and large because the problems, the environment doesn't always work. But I do think what we have to do is work at harnessing the collective expertise that exists in universities. And I'd like to see governments, maybe on a continent scale, provide incentives for laboratories to work together as teams instead of just doing their own individual research. Because all the big research these days is done with a team. It's very rare that a single laboratory is going to make a breakthrough, and find ways to bring together people with complementary skill sets who can take a creative approach to a particular problem. And again, focus on African issues. It was just mentioned about the genetic diversity in Africa. So one thing we don't even know is will the African population respond in the same ways to the drugs developed for Western or Asian populations? It's an area that's ripe for study. Also, you mentioned, it was mentioned earlier that we don't have to have the newest drug. I couldn't agree more. In fact, one of our projects involves studying old cancer drugs in this particular case that are orally available and that could be used to treat cancer patients in rural settings where setting up an infusion center is just not possible. But we have some very good cancer drugs, and we've explored some. Some are quite toxic, but there actually are a few that are oral and could be used to extend the life of cancer patients with certain types of cancers. And I think if more people got involved in those kinds of undertaking, looking at older drugs and that are very inexpensive, and especially oral drugs, and seeing if we could utilize them to improve the health of especially rural populations. I think there could be many opportunities. I fear we could talk about possibilities for hours, and I know we don't have hours, so I'll stop talking at this point. But I want to thank the other members of the panel, because I think you've provided us with a lot of great insights.
Thank you, Dennis. I think the final word will go to Tony, but that's not the final. The House gets to have the last say. And unfortunately, the House is shared between myself and Lawrence. So if I could just get your final word, Dr. Ndefo, especially because when Dennis mentions mining old cancer drugs, I have a feeling that this is one of the areas where the infrastructure, the shared infrastructure that you spoke about, could become very, very critical, not just for the old cancer drugs, but even for biosimilars. Over to you, Tony.
Yeah, very true. And that's also an area that, pharmaceuticals, such as big pharmaceuticals, such as J&J, have worked with many companies, many countries to support that effort in terms of sharing their libraries or allowing researchers to mine their libraries for compounds that can be used for various other indications. And I think, that I've worked with the colleagues in South Africa who've actually been working on that in Cape Town, who've been trying to do that. But I think one of the things I want to touch on is just going back to innovation. I think one of the things that we really do need, outside of the standard innovations in the lab, is really innovations in collaboration. And I think that's probably, we've talked about it, how between country, across regional collaboration, I think that becomes very, very critical, because for any one of these things that we're talking about, because some of them end up being very, very resource intensive, it's not the talent that is lacking. The talent is clearly there right across board. And but what we need is to figure out different ways, new models of how we can collaborate and tap into various regional centers or even countries or expertise across countries, and even the diaspora, and how we can tap into them to make many of these innovations, making of this many of this work. There isn't an absence of talent right across board, and but I think figuring out how we can collaborate is really essential to get this across, and also not to pander on Rich's comment about African dictators, because I think there are equally, if you look quite in the east of Europe, there are equally as brutal dictators, but but there are unstable governments, right? And that's a fact. That's a fact that for any research to work, you need stable environments where you can ship things and they can go to where they're supposed to go. You can get items with appropriate taxation. So there just needs to be some consistency in a way that things work, some predictability in a way that things work, and that is something that is lacking in several countries, and many countries are doing an excellent job in trying to fix it, in sub-Saharan Africa in particular, but also there is clearly a need for some innovation there, as we move forward.
Thank you for that last word, Dr. Ndefo. As I said, the House gets to have the last word, and sitting with me in the House is Dr. Lawrence Banks. I just want him just to close, particularly in regards to what he mentioned and what several of the panelists have said as well, which is the first prize is to provide medicines, to provide adequate health care without worrying about innovation, whose sole objective in the way of creating Christensen is to extract more profits. How does he see that in terms of the ICGP equation?
With ICGB, we want to make sure that any of the technologies that is transferred, whether it's from us or from elsewhere, we have an arrangement with the people on the ground, the companies that are going to be producing, that they obviously, I think Richard's model for NEB, saying you start small and build up, is absolutely perfect. You make enough money to survive as a company so you can make investment and develop new products or new procedures, but you're not aiming to be a global superstar. Because if you do that and people are just driven by the money, it's not going to get to the people that need it. So I think that would be the main thing, that you set that in place right at the very beginning when you identify the players who are going to be your manufacturer, your producer. You ensure that you have facilities in place that mean that they're going to market it at a level which is sustainable. And I think that's absolutely fundamental. If that's my last word, one other thing I want to say, and that's to say to Nhlanhla and the whole team here at BioAfrica, a really, really heartfelt thanks from everybody for doing this. It's such an amazing convention, and from all the talks that I've sat through, I've learnt so much, and the collaborations and the list of contacts I have that I need to follow up from this meeting is going to keep me very busy for many weeks. So thank you so much indeed.
I think with those words, I would also like to thank my esteemed panel. Thank you for taking the time to share with us your wisdom. Thank you for making, without knowing, commitments that I am intending to follow up. And I think BioAfrica as a platform has been designed, and we hope to grow it further to try and create and conceptualize solutions in Africa for Africans that are driven as a goal to deliver healthcare and food security to the continent, driven by the Africans themselves. And I'm very, very grateful to the panelists, to the delegates, to all the speakers who have contributed so much over the past 2 days. I am grateful beyond words, and I'm pretty sure the team at AfricaBio is very grateful. And at this point, I would like to hand over to the Deputy Chairperson of BioAfrica Convention, Dr. Tumi Simete, to close the convention. Thank you once again.